So this is the title of my talk, also the title of a paper that's coming out in PLOS Genetics in a couple weeks. So you're getting a little bit of a preview here. And I've listed myself and Peter Kloss on the cover here. There are 20 other people as well who have all contributed substantially, but I'm doing this mostly because this is really Peter and my project together, and we're moving forward in a lot of directions, and we're really excited about this. So let me start. Let's see. I push this arrow here. Yeah. All right. So molecular photo fitting is a term that Tony Fredakis and I came up with, I don't know, maybe 10 years ago. And it's the process of predicting phenotypes from a DNA analysis. Actually, we stole the Forensic Science Service DNA photo fitting. Other people call this molecular montage, and, you know, variety of names. There are two basic types. Indirect molecular photo fitting, where you're using ancestry. So you predict ancestry, and ancestry is related to a lot of traits. So you can learn something about a person that way, an unknown suspect. And then there's direct molecular photo fitting. This is working to assay the genes that actually functionally affect the trait. Okay, and so that's what we're working on now. And the hope is to get something like a driver's license. You know, the information on there. You have the height, the eye color, a picture of the person, a snapshot, you know, of their face. Even two of them now. Sometimes there's even one at a different angle if you look at some of the more advanced biometric IDs. But these, there's a whole series of traits. Some of these are pretty easy, like sex, pigmentation. We're quite a bit of the way there. Others, like hair texture, there hasn't been a lot of work done yet. And facial features really has been one that has been really hasn't seen much progress, even though we do know a lot about many aspects of the developmental biology of the human face. So when would you use this? You know, I think this is an important issue to address up front, because this is going to affect how people take in the information, and you know, what kinds of controls there will be on the precision of the information and when it's ready really to be used. So I made this little this little plot here where you have the evidentiary sample and then a reference sample. The reference sample could either be a suspect or a database, like CODIS for DNA, or for facial images it could be the driver's license database or passport database, INS database, these kinds of things. So if you have evidentiary DNA and reference DNA or a database, you use STRs. Right, there's no question. I mean, SNPs are coming online too, and there'll be other molecular methods as well. If you have a facial image as evidence, a facial image in the reference, you use biometrics. And there's a whole bunch of different methods that you can use to compare faces. Most of them really use a 3D model as well. So you take a 2D image or a video image, predict the 3D model of that person's face, and then compare those 3D models, because you can adjust for pose and scale and those sorts of things. But what we're talking about with molecular photo fitting is either having as evidence DNA, but no DNA to compare against. So we're trying to create a model of what the person might look like. And then compare that against facial images from a reference. In this case, it could even be those facial images stored in people's brains. You know, do they know this person? Do they recognize this person as somebody they're acquainted with? You also have, you know, evidence as a facial image. And if the right DNA database exists, you could compare that facial image to the DNA database. So those two go hand in hand. And of course, you know, things like craniofacial reconstructions. I'm sure there's a bunch of other things that aren't really standard forensics either that these methods can be used for. So this is kind of a complex chart, but really when you're thinking about traits like facial features, you have to remember these are really complex traits, and they probably are subject to lots and lots of different genes. I think there's over 400 craniofacial anomalies. So these are, you know, very obvious changes due to strong mutations in particular genes. So that's the first one here, craniofacial disease mapping. Normal range mapping, that's a lot of what we're doing and what I'll be talking about today. Sex-dependent gene mapping, there are distinct differences between males and females of the human species in their face. You have to remember too, humans differ sexually or across the sexes in ways that not all other apes do. There's a lot of unique aspects of the evolution of our species and other species, in terms of sexual dimorphism. And sexual selection is definitely an important force in guiding those differences. Animal models, there's a bunch of mouse models and zebrafish models, other animals. I mean, just look at dog breeds, right? Lots of different faces. Gene expressions, there's a lot of molecular biology and some functional genomics. And finally, population genomics. This is one of my favorite things. This is tracking the evolution of each part of the genome and trying to say, well, does that region show an accelerated pattern of evolution? Most of the genes I'm going to talk to you about today, we screened for that because we were looking at an admixed population and we wanted to increase our chances of the candidate genes that we knew about playing a role. So we looked at those candidate genes that were different between the two parental groups. So they showed a pattern of accelerated evolution because, populations differ. And if they differ in a way that's caused by this particular gene, then that gene has to have a big difference between the populations in terms of frequency. Okay, so you discover the genes. You validate that discovery. That's an important step. I'm not of the opinion that you can just use millions of markers and just go at it without using all this other information. I think, there's too much to learn and too much that we already know that can augment that. Because you want to use, at least our approach is to use a focused set of information. You don't want to be modeling noise. Some of these methods are so powerful that you'll get results even when they're not statistically significant. So that next step, that orange box, is the population database. So this is the larger set of individuals where you have measured lots of things. Some of the things are these genes, these facial feature SNPs. Some of the SNPs are also AIMs. These could be STRs too. The typical STRs we use in forensics don't have a lot of ancestry information, but there are STRs that do. But still, SNPs are easier to type, and if you're already typing SNPs, just type ancestry SNPs. And then you have some self-report or measured features, things like sex, age, body size. So those, along with the AIMs, we model independently those effects, because those are strong effects that sometimes don't even have to do with genes. Age, for example. Your genes aren't changing that much through your lifespan. You're just, developing and then aging. And why do we think faces are genetic? Well, there's a lot of reasons. I mean, identical twins, really strikingly similar faces. Family resemblances, you see some of that in this picture even. Population differences, those can be distinct. Those can also be exaggerated in our minds. I'll show you a little bit of that too. Sex differences, I think we diminish these too when we observe individuals' faces. And craniofacial anomalies. So here's our approach. We use a quasi-landmark approach that Peter Klaas developed. Actually, first he developed this for craniofacial reconstruction. He has CT scans in his PhD, so he could see the underlying bone structure as well as the surface structure, and was able to use that tissue depth from CT scans in order to put faces on skulls. So, but we use it to start up here. You probably can't see my mouse, right? The cursor. But face A there is the original. Okay, there we go. A is the original face. B is that face just trimmed. We got rid of the extraneous parts. C is the reflection. So to reflect a face, you just have to multiply the x-axis by -1. So you're just flipping around one of the three axes that define an object in three-dimensional space. Then we map this anthropometric mask, which is D. We map that onto the original face as well as onto the reflected face. So we do that twice. Then, for the purposes here, we've just gotten rid of the asymmetry information. Asymmetry is quite fascinating as well, and we'll be looking at that and looking for genes that cause different patterns of asymmetry. We've already published a paper on the differences between males and females in the patterns of asymmetry. There's probably differences across sexes, or populations, there's probably differences depending on genotype as well. But here we just average those, and to do that you simply average the x-axis variable, because we're talking about bilateral symmetry, so left-right symmetry. And H is what you see there at the top as what we call the projected face. So we do a PCA on all those G faces, all those symmetrized faces. We put into a principal components analysis and get however many components that explain what level of variation we want to explain in the model. And H then is just that subset of components that are used to reconstruct that person's face. So these are the first 5 principal components. The data set I'll tell you about today has 592 people who have mixed African and European ancestry. So these were drawn from 3 countries, Cape Verde Islands off the coast of Africa. People there are about half African, half European, and many generations back in their mixture. African Americans and some European Americans who had enough African ancestry that we would be interested in using them here, and finally Brazilians. So, we used a cutoff of 98%, which is typical in computer vision work. So that gave us 44 PCs. So not an infinite number of PCs, which is really nice. And you can see the first PC is largely a sex PC, right? You got a female face and a male face. The second PC is both sex and ancestry, and you can see, the brow ridge, as I'll show you in a minute, is something that differs, of course, between the sexes and really mostly in European populations. But it also differs between ancestry groups, at least these 2 ancestry groups. So European populations have much stronger brow ridges than African populations. And then you get to more specific parts. Again, you're seeing some ancestry and sex, right? The face on the left here has both male appearance and more African appearance in this face here, more feminine and more European. And then you also get to particular parts of the face. We could go through this whole list and find interesting aspects of each of these PCs, but I just want to give you a flavor of those PCs. And here's how a few populations pan out when you use just the first 2 PCs. Here are the names, and I split males and females separately because they do tend to cluster separately. There is a lot of sex difference between the populations. So you can plot them like this. What we're working with, though, is really a multi-dimensional space. So it's easy to show the first 3 dimensions, but once you get to the 4th, the 5th, and the 44th dimension, it's much harder to imagine. But the math is not any more complex. All we do is, since the principal components are independent by definition, this is how they're calculated, you just sum up the results. So you make each calculation for each principal component and add them up, and then you have the joint effect of all of these because they're all independent. You see some interesting factors here. You might think, well, I'm going to lose a lot of data if I use just 44 PCs. With a sample like this for 592, there are actually 591 principal components. So we're throwing away most of that. And some of it we definitely don't want. If you look at these 3 people, the scan cleaned face is on the top and the remap face below it. It's very clear that the person is the same, and we're losing some of the artifacts, like this beard here on this first guy, some bangs on this second girl, and some really big bangs here on the third girl. We are also getting better at moving the hair out of the way and taking better images of the ears too. Ears are quite fascinating, and Peter's got a paper he's working on right now with ears. You know, you can clean up the data actually by using a subset of PCs. And Peter and I also developed a new modeling method, a new response modeling method that we call bootstrap response-based imputation modeling. It's actually pretty cool. We force an imputation. So we leave out each person in turn and use the rest of the people as the training set. And then we take that value that we give that person, which has nothing to do with what his value was originally. It has everything to do with what his face would represent his value to have been. And we use that as his actual value and then proceed. And we do this in an iterative fashion. The paper goes into extensive detail on this fact because it's a new method, but we put all that in the supplementary material, so not everybody has to wade through it. We can then, using this method, BRIM, we can recombine all those independent PCs. So each of those PCs has a chance to have some effect on the trait. And we allow that. You know, we use partial least squares regression, which is a way to compare sets of matrices to each other. Then all those PCs can each have a new effect on the face, and we summarize them into a single number. So this is showing the facial ancestry, or RIP-A, the response-based imputed predictor variable for ancestry, against West African genomic ancestry. And this genomic ancestry is derived just from 68 AIMs, 68 SNPs. So it's not a very informative ancestry measure, and in fact, the facial ancestry is much more informative. So this facial ancestry was derived with those 68 AIMs. Could one of the organizers tell me how much time I have? Like 10 or 15 minutes? Yeah, I'll just work with that. So this is that same plot, but I'm showing you here which are the three populations, which are the ancestry groups. So the Brazilians are down here in red, the Cape Verdeans in blue, and the Americans in black, both at the top and the bottom. This is a plot showing how we get much more information. This red square over here, this big red square, you know, is the correlation between the RIP-A estimates. And some of these were estimated with skin color. So M is skin color here. 3 is just 3 AIMs, 15 AIMs. So you can have very low levels of input information. And when you BRIM it in the end, you know, you get a 98% correlation. So you get really high correlation, even if you use very little ancestry information to begin with. So it's a very, very powerful method. It then allows you to select faces at the extremes on that RIP-A variable, for example. So here we didn't average together African faces and average together European faces to get these, which is possible, and I've done this and other people do this, but this is using that RIP-A variable that we derive from there. We can just, so it's a direction through this multidimensional space. Each of those 44 principal components is affected to some extent by ancestry. And so the RIP-A is a measure of that. And you get faces that are clearly, you know, African typical, European typical. We also came up with a new term and a series of different ways to represent the differences between two faces, and we call these face shape change parameters. Typically, people will plot X, Y, and Z in these temperature plots, but it's really hard to imagine unless you look at a lot of these what's actually changing with those. So we instead worked on the normal displacement. So a normal plane is like a tangent line to a curve. So it's that plane that best fits a flat edge of that shape. So this is how the normal shape is changing. So the brow ridge is going out, the nose is going out, the lips are going in. What you're seeing on the top there. Then the area change. So how much are those polygons changing? that connect those, and this tells you how the surface area of that face part is changing. And then finally, the curvature ratio, and you can see here the biggest change between West African and European is under the nose where the philtrum meets the columella. Europeans are almost convex there, whereas most African individuals have a concavity. And you see the brow ridge here too. I mentioned that before. So Europeans have a stronger brow ridge that's coming out in that plot. So we can do this for sex too. And you see, again, you see the brow ridge as a big effect. You also see the nose bridge. So because the male brow ridge is much stronger, you get this curvature here, and that's more enhanced in European populations as well. So let's see. So when we compared these results to a lot of different things, like the actual sex of people, that's what's shown here. So the RIP-S value differs substantially between males and females, more variation in males. This result here is pretty cool. This is showing the misclassification rate. So we can actually call a third of the sample the wrong sex. Just make 15% of each males and females, just flip them and say, code them wrong, and still get 95% correct classification by the area under the curve. So it's very powerful at reclaiming the patterns. I'm going to skip this. This just says that facial femininity has a much bigger sex difference than height, another trait that we know is very sexually dimorphic. Then we can transform faces and do this also with the texture map properly assembled. So here I often ask people to tell me which of these is the real face, and they all look pretty accurate. I think the actual person is around here, but we've just transformed him, made him more African going down, more European going up, more feminine going to the right. And we've done some work with the perception of these traits. I think this is really critical in understanding both how we see faces and what psychologically is affecting those perceptions, as well as giving us some good additional scores to compare these things like RIP-A and RIP-S to. So here you can see a very strong correlation between ancestry and or between RIP-S and the ratings of ancestry and judgments of ancestry. And same thing with sex. I mean, there's some interesting patterns here, and we're going to work this up into another paper. Some are summarized here. There is more sex variation than ancestry variation, but people don't see that. People are more focused on the ancestry variation. People also see male faces as male much better, and I think there's probably an evolutionary reason for that. You want to avoid those. I mean, look in the prisons, right? You got a lot more men there. We can use these same methods to model individual genes, and that's one of the points of this paper. So this is showing a particular disease, Smith-Magenis syndrome, and you can see you get this tented upper lip. The philtrum has that appearance, and our model of the normal variation has that. Also, the nose is upturned in the craniofacial condition, and our model shows some of that pattern. So we want to, in this paper, we just did subjective assessments of the appearance of these traits. We want to do this subjectively. Here's another one. This gene, LRP6, it's not known to cause a human craniofacial anomaly, but when you knock it out in the mouse, you lose both lips. Mice have a split lip, so there's actually two lips, but you lose both of them. And in the human range, look at the lips on this. So you get a really enhanced difference between the lips from the normal range effects of these genes. We're releasing this program, DNA to Face in 3D, that lets users or readers explore the effects of all of these. And finally, I want to present just a few reconstructions that we're doing. This will, again, be a further paper, but I wanted to share some of these because I think even at this stage, and I want to really qualify this strongly, these are methodological proofs of concept or proof of principle. We're showing we know how to do the prediction. We know how to assemble all of those genes together into estimates of the final face shape. And we do this by first making the base face from sex and ancestry, then adding the SNPs on, and finally we exaggerate them. We're boosting them here for the human observer. Humans see caricatures of faces much better. One thing that inspired us is this Japanese TV documentary, so Peter and I got involved with them, and this is the real face on the left and the predicted face on the right. And this one worked pretty good. The chin's nice, and the nose, I'm not so sure we captured too much. We can add age as well. Most, our model's not really well adapted or not well selected, the sample of people. Here's another face from that study, and here's some more pictures of her. I think this one came out pretty good. A small chin, a bit of a wider nose. Here are the projected faces, this one from the PCs, and this is the predicted face down here with the texture map and without. And I just want to show you a few more. This guy's a bit older on the top when I have his actual picture. I've seen pictures of him younger, and I mean, he has this really spectacular nose. He's actually upstairs right now. This is my father-in-law. And, I think we captured a bit of that here. So, I mean, maybe we're picking out traits just because they kind of strike us, but I just want to show you a few more of these